Hello, everyone. Last week we talked about coliforms and our different methods of enumeration, including our violet-red biologger, VRBA, and our simplates, which is our statistical enumeration method. Now here we have IMVIC, which is our method to differentiate between our pathogenic and our non-pathogenic coliforms. Right, E. coli is our pathogenic and Enterobacter is our non-pathogenic. Today we're going to be discussing Staphylococci, which is a group of bacteria that are based off of, their name is based off of how they look underneath a microscope. So they're going to be gram-positive, so if you stain them with a gram stain, they'll be the violet color. And they will be forming bunches or clusters, which is what the Staphyla part of the name refers to, bunches of grapes, and then the coccus is the shape of the cells, which, as we discussed before, coccus is the round shape circle, and then the bacillus would be the rod shape. And the Staphylococcus, this genus, is often found on the skin or in the nose, and because of that, it's going to be an indicator of contamination from food handlers. So anyone that handles the food and is going to be touching it or possibly sneezing on it or something like that. The most pathogenic form, one of the most, is Staphylococcus aureus, which is also known as gold staph, and a major virulence factor that is produced by Staph aureus is coagulase, which is a cell surface protein that is going to convert fibrinogen to fibrin, which is going to form clots inside the blood, which for obvious reasons we really don't want, because that can cause heart attacks, strokes, lots of not good things. Then we have heat-resistant enterotoxins, which will not really be broken down by food, or by heating your food, hence the heat resistance, which is why they're such a big deal. And that can lead also to gastroenteritis. Now, any food that has greater than or equal to 10 to the 6 cells per gram of Staph aureus are considered dangerous for consumption. This can be caused by poor food handling, and especially for foods that aren't cooked, such as salad or sushi. And then if a food stays at room temperature for a long time, the small amount of staph on there, Staph aureus, will multiply over time, and eventually you can get them to above your dangerous levels, and then it won't be safe to eat anymore. So be sure to be careful of keeping food at room temperature for a long time for that reason. Now, coming up next are some graphic images, so I just want to warn you ahead of time. Here we have examples of staph infections. So these are all on the skin. A few different symptomology and types of disease, but the people who are mostly at risk are going to be people who have weakened immune system, so whether that's from an acquired immune deficiency, or if you're going to be a newborn, an elderly person, or a woman who's breastfeeding, all of these are going to be people who are highly at risk. And the signs and symptoms of this infection is pain, redness, pus, swelling, and then fever, chills, and low blood pressure as well. There are a few different types of diseases: mastitis, pneumonia, sepsis, impetigo, and cellulitis. For our methicillin-resistant Staph aureus, which is methicillin-resistant, methicillin is going to be an antibiotic used to treat Staph aureus, and so this is known as a superbug because it is resistant and is going to be a lot more dangerous and going to cause more severe infections and is a lot more difficult to treat because of its resistances. Now, because of this resistance to methicillin, it's going to be very common in healthcare associated situations. So the symptoms are very similar to what we were looking at before, again, but just a lot more severe. And you can catch this by sharing any objects that came into contact with skin or any skin-to-skin contact. The other species of Staphylococcus is Staphylococcus epidermidis, and this is your non-pathogenic species. This is going to be commonly found on skin and mucous membranes, very similar areas as the Staphylococcus aureus, but since this is non-pathogenic, we don't really have to worry about this causing any infections in humans. Now, just like when we were differentiating between our coliforms, between our pathogens and our non-pathogens, we also want to be able to differentiate between our pathogenic Staphylococci species. So in order to differentiate between Staphylococcus aureus and Staphylococcus epidermidis, we can perform our coagulase test, because Staphylococcus epidermidis does not have any coagulase. And then we also have our thermonuclease production. We can test for that as well. So these two things, as well as mannitol, the utilization of mannitol in anaerobic conditions. So these are a few different ways that we can differentiate between our pathogenic Staphylococci genera. Now, if we were in lab, we would be performing a gram stain and then a streaked isolation on Baird Parker and biochrome Staph aureus agar. We'll be talking a little bit about that more next week. And we also would have been placing a drop of rabbit serum, which is blood cells from a rabbit, and we're going to be placed that on a slide, and then you mix that together with some of the cells, and if the cells can create coagulase, then the blood will begin to clump and you'll see clumps in the blood. And then in order to test for catalase, you can place H2O2 on the colonies, and if catalase is present, then you will have bubbles appearing on the colonies. So that's it for the PowerPoint portion for today. Make sure that you are going to take a look at the rubric for the sauerkraut report. There's a lot of useful information in there that'll really help you to figure out what exactly I want from you and how to get the maximum amount of points for this report. It's just going to be a PowerPoint between 5 and 15 slides. I just want you to talk all about your sauerkraut making process, how you made it, and all of your observations that you had throughout your process of making your sauerkraut, and everything that I had in the rubric. Just take a look at that. That'll really clear up any questions. And hopefully, if you do have any questions, you will email me and let me know. And I will try my best to help you and answer as soon as possible. Thank you, everyone. Good luck.